This video is for scientific, academic, and educational purposes only and does not constitute medical advice. In 1980s Bologna, Italy, a 52-year-old physician experienced a sudden, irreversible shift. He could no longer fall asleep. As the condition took hold, researchers utilized vintage EEG monitoring to track his brain activity and attempt to identify the cause of the onset. For 18 months, the symptoms progressed without respite. The insomnia was absolute, resisting every known clinical intervention. This prolonged state of wakefulness eventually triggered a total collapse of the autonomic nervous system, leading to the patient's death. Polysomnography data from this period reveals the severity of the deficit. Where a healthy brain generates smooth, rhythmic delta waves during deep sleep, this patient showed only chaotic, dissynchronized wakefulness throughout the night. The patient's brain had physically lost the biological capacity to initiate the state of sleep. Doctors initially attempted to force the brain into rest using high-dose sedatives and continuous barbiturate infusions. While these chemicals could render the patient unconscious, the underlying EEG data remained unchanged. They failed to induce restorative sleep. Sleep is an active, highly orchestrated neurological program. It is a biological state that requires specific functional hardware to execute. The center of this program is located in the diencephalon. The bilateral thalamus acts as a subcortical gatekeeper, responsible for generating the brain's sleep architecture. The Bologna case was eventually identified as the first modern clinical record of fatal familial insomnia, or FFI. FFI physically destroys the neurocircuitry of the thalamus, specifically the regions required to generate sleep spindles. Because the structural hardware of the thalamus was gone, no amount of chemical sedation could ever generate slow-wave delta sleep. To find the origin of this thalamic destruction, the investigation must move from macroscopic anatomy down to the genetic level. Fatal familial insomnia is an autosomal dominant prion disease. It carries a lethal penetrance of nearly 100% across a family lineage. The source of the disease is located on the PRNP gene, found on chromosome 20. The destruction is triggered by a precise point mutation called D178N, which substitutes aspartic acid with asparagine at codon 178. For the disease to manifest as FFI, this mutation must be coupled with a methionine polymorphism at codon 129 on the same allele. In a healthy state, the PRNP gene produces PRPC, a cellular prion protein that maintains synaptic health along the neuronal membrane. This specific coordinate, D178N paired with 129M, is what transforms this routine maintenance protein into an unstoppable neurodegenerative weapon. The mechanism of this transformation is found in the baseline geometry of the healthy PRPC protein. Normal PRPC is composed primarily of soluble, winding alpha helical coils. The D178N129M mutation triggers a catastrophic error in the protein's folding process. This severe error causes the soluble alpha helices to undergo a violent physical collapse into a rigid, misfolded shape. This newly formed abnormal isoform is designated as PRPSC. The protein's architecture is now dominated by interlocking, jagged beta sheets. Human proteases, the enzymes responsible for breaking down misfolded proteins, are physically unable to digest or degrade this beta sheet structure. This genetic mutation structurally engineers an indestructible protease-resistant barricade within the brain tissue. Prions are a biological anomaly because they lack DNA or RNA, yet they propagate through the brain with aggressive speed. They spread through a mechanism called template-directed propagation, where the misfolded pre-PSC acts as a physical stamp. When a misfolded pre-PSC protein collides with a healthy pre-PC molecule, it physically forces the normal protein to snap into the pathological beta sheet shape. This creates an accelerating chain reaction that moves across the neuronal membrane. These indestructible beta sheets eventually aggregate into dense, insoluble masses known as amyloid fibral plaques. As these dense plaques expand, they physically destroy the surrounding neurons from within. This exponential, template-directed aggregation is the direct cause of the systemic failure observed in the Bologna patient. Zooming out from the microscopic plaques, we see how this destruction manifests across the entire brain. Creutzfeldt-Jakob Disease, or CJD, is another lethal prion disease that serves as a useful comparative baseline. While CJD causes widespread damage across the cerebral cortex, FFI exhibits a chilling anatomical selectivity. The D178N129M variant targets and destroys the mediodorsal and anterior ventral nuclei of the thalamus with surgical precision. Researchers are still uncovering why this variant targets these exact nuclei, but the clinical result is absolute. These specific targets represent that neurological hardware that the human brain requires to generate the biological state of sleep. Because prions lack nucleic acids and their beta sheet structures are indestructible, the codon 178 cascade results in the permanent, fatal dismantling of the human sleep-wake cycle.